is we need to finish off the last question from the homework discussion. I think we have one more to do. Talk about myasthenia gravis. And then we will be actually, at that point, we will end the unit as far as what will be on the upcoming exam. And we're going to start in today on Chapter 7. So slides for Chapter 7 went up, I don't know, like Tuesday or something. I think I forgot to post an announcement about it. But we'll be starting into those. 7's a little bit different in that there's sort of there's a lecture, there's like a chapter proper for Chapter 7 that kind of gives an overview of anatomy of nervous system. And then all the nitty-gritty details are in the appendix. And I've kind of done the same thing with how I'm presenting it. So I've got a lecture for the chapter part. And then there's a worksheet that I've also posted that'll be more for focusing on the appendix. So we'll start into that today. Wednesday we'll continue onto that. I think there will be time to work on the worksheet in class on Wednesday as well. The worksheet itself is going to be due on Friday. But again, we'll get into that in a second. Monday, I'm sure the big news, or the big topic of focus is the exam. So exam number 1, midterm, is going to be on Monday, same time, the same place as usual. Nothing unusual about it, except we will be doing the exam. You'll have, I will try to be here by 1 o'clock if you want to start a few minutes early. So at that point, you could start 10 minutes early if you come at 1 rather than 1:10. But we do have to be out and done by 2. We've already talked format a little bit in here. Do you guys have any questions about the exam or anything? There's a study guide that's on under Module 1. I think that can be useful and helpful as a way to sort of work your way through the slides. But please use your lecture slides, your notes, your homework as your main study resource, and do it. The rest of it should be supplementary stuff. If there's stuff that's in the chapters you do not need that we did not cover in any way on homework or in the lecture, then you don't need to know it. Any questions? You guys feeling okay? Get some nods. I'm not getting any like massive deer in headlight. Kind of like just like no. So that's good. Okay. Also, I just want to say, please feel free to send me questions. I mean, we're kind of getting towards crunch time here, and I know the weekend's kind of right before the exam, which is not always my favorite way to do things because I'm less accessible. But I definitely will be around my email this weekend. So if you have questions, please feel free to send me questions. Ideally, they would not be, can you please explain all of Chapter 5 to me? Because then I would have to write like 15 pages to explain all of Chapter 5 to you. I probably will direct you to go read the book at that point. But if you have I don't understand this slide, or I don't understand this specific topic within this chapter, or, whatever the thing, or I don't understand this question on the study guide, send me that and I should be able to, help you. Maybe don't send me 20 questions per email, but I will try to help you as best I can. And I'm happy to respond, okay? All right, so the last question we had, let me flip back over to it. Here it is. So we were talking Chapter 5 questions. The last question is about myasthenia gravis, which I know is kind of like in a different section. I had you read box 13 too, and you're probably like, what the hell, we're in Chapter 5. But it's really directly relevant to this because it all has to do with what's going on in the synapse and how, as far as what's going on with this disorder. So the question asks you to report what are the disease's symptoms, what is its pathophysiology, so what goes wrong specifically to cause those symptoms, and then why it would be helped by acetylcholinesterase inhibitors drugs. And I think, Emily, you had it, right? Okay. Okay. Scroll up and turn the lights so you can actually see. Anybody have anything different or additional they would want to throw in for any of these? I'll be happy. I'm giving you a second because everybody's madly typing, so I'm letting you madly type. To me, I think we're pretty much spot on. So myasthenia gravis, like it is this issue of extreme muscle weakness. So this could be sort of manifest in just sort of fatigue or inability to, you feel like you can barely move. A lot of times you see people's eyelids kind of get droopy because they start to lose muscle tone in the muscles that are controlling your eyelids. And just as sort of one of the more immediate kinds of symptoms that you realize early on, it can progress and become worse and worse. That's not quite how you spell symptoms. It is an autoimmune disorder. Definitely you have you have antibodies that are attacking the acetylcholine receptors. There's actually several things that can happen as a result of that. So literally one thing that can happen is if you have your receptor sitting there. So if my hand here is the receptor, and you have an antibody that comes in and binds to it, it can just block that receptor, and therefore acetylcholine can't get in. Another thing that can sometimes happen is that the acetylcholine receptors, because they've been bound by an antibody, the cell might just then start to pull them in because somehow it detects that they've been damaged, and so it kind of pulls them in and processes them. But either way, those acetylcholine receptors are not available anymore for actual acetylcholine molecules to bind to them. And I think C is also, I think, is a nice way to put it. So acetylcholine esterase inhibitor. Acetylcholine esterase is the enzyme that breaks down acetylcholine in the synapse. So if you inhibit that enzyme, that's going to result in acetylcholine just lasting longer in those synapses. And so you get these sort of prolonged stimulations. Like normally this would result in hyperexcitation of your muscles. It might even result in sort of your muscles kind of being overstimulated. But in the case of somebody with myasthenia gravis, when you deliver this or provide this medication, this in the short term can result in a relief of symptoms. Unfortunately, what seems to happen is that if you continue to administer it over long term, you have sort of an acclimation effect where I'm not sure if you just start making more of the enzyme or something happens where it loses effectiveness after a time. And so it is not a long-term treatment option, but it is an option that people will use as sort of a diagnostic procedure, at least, that if they can fix your symptoms by giving you acetylcholine esterase inhibitor, it is making it more likely that this is your problem, that you have myasthenia gravis. It's consistent with those symptoms. I don't know if it would just be, why it would just be one. I mean, I just have to imagine one set of cells is getting affected more rapidly than the other. But I don't really know. Other questions? Yeah, good. Oh, yeah. Well, here, let me do a thing. I can't do that. Other questions on myasthenia gravis? All right, cool. So again, I think that's an interesting disorder. We'll talk about, I try to hit on other major neuromuscular disorders as we're sort of working through relevant parts of the process. And certainly this is one where you've got a problem in the synapse, so it fits nicely within this chapter. Okay, well that ends the unit. So we are done with Unit 1. We're done with the material for Exam 1. So what I want to do for the rest of today is start into the next unit and just sort of introduce that. So Chapter 7 is about the structure of the nervous system. And there's a way in which I feel like Chapter 7 is almost like development of nervous system, because we're going to do sort of a story about the development of how the nervous system develops as a way to understand the structure of the thing. And I think it actually provides a really nice framework for understanding it, because otherwise there's, especially in brain anatomy, the brain gets kind of contorted and twisted around in different ways, but it really is kind of a linear structure. And if you focus on the development of it, I think it helps you understand that a little bit better. So we're going to start with discussion of the development of the nervous system. There's a process called neurulation that happens very early on in embryonic development, and then you have differentiation of structures, the differentiation of the neural tube, which gives rise to all of your central nervous system. And the neural crest, which is going to give rise to all of our peripheral nervous system. And then after that, we'll kind of just do a survey of important brain centers. That will set up then what we're doing with the appendix. So the appendix for this chapter is just like a nice foray into neural anatomy. So that will go through in much more detail different parts of the brain, a little bit on spinal cord as well. Cranial nerves are nestled in there. So there's lots of anatomy. And I'm not really an anatomist, so we're going to do not all of it, but a good chunk of it. I think enough that we have a good basis for what we'll be doing afterwards as we sort of move forward through other chapters, because we're going to re-reference a lot of the things that a lot of the structures we'll be learning about in this chapter as we move into trying to understand how our somatosensory system works, how our motor systems work, etc. You might remember this flow chart. This is kind of giving you an idea of where we've been and where we're going. So I put this in chapter 1. This is kind of my organization for the discipline of neuroscience as well as this course. We've been kind of on this part where we've been looking at cellular structure and then cellular function and trying to understand how these things work. So, what's going on at a cellular level and how do cells communicate with one another? How does that whole process operate? What we're doing in this chapter is kind of setting up nervous system structure, so we'll try to understand how the cells are assembled. And then once we have all that, once we understand how the cells work, and once we understand how they're all assembled into the larger structures of our central nervous system, then we can understand and focus more on function behavior, and that's where we'll be the rest of the semester. So we'll be looking at these sort of loops between sensory input, integration of that information, decision-making, and ultimately motor output. And then, of course, this is one big cyclic kind of a process. So to best understand nervous system structure, I really like what your book does in that it kind of walks you through the development of the nervous system. And I think, we're not going to be getting into much of like the molecular signals and all the really hardcore developmental biology. This is meant to be more of like a descriptive story of how the nervous system develops. But I think it's a useful framework to understand the nervous system better. So pretty early on in development, this is well past the point of fertilization. So to get to the point that I'm trying to depict on this slide, we've already had sperm meet egg. We've had sort of an initial round of division where you kind of just create this morolla, which is just like a little ball of cells. It looks a little bit like a berry, a really small berry. And then that ball of cells becomes a hollow ball of cells as all the cells kind of get marginalized to the side. You get finally into the uterus, you implant into the uterus, and that's what we're seeing here. So we have an implanted embryo. It's kind of hard, I cropped too much of this picture, but this is like wall of uterus that we're seeing through here. And the embryo would have landed probably would have landed here. The wall of the uterus used to be around here, and the wall of the uterus has kind of grown around this embryo. But the embryo itself is this little structure in here. All of this out here is going to become ultimately the placenta and surrounding supporting embryonic tissue. And you can see if you look in here, the embryo is composed, there's sort of like a ball of cells again, but these are still, again, more sort of like supporting membranes that are not really going to become part of what's going to become this person later on. In that you've got, you've got the amniotic fluid or amniotic cavity, which is right there. You don't need to know that, but just so give you some reference. So this is the amnion, and this will eventually grow all around. This whole thing down here, this is the yolk sac. So that tends to be, it's sort of prominent right at this point. It becomes less prominent as we move on and all the other structures get bigger, and we don't do much with our yolk sac as mammals, but of course that's really important for birds and reptiles and such that lay eggs. The part of this developing embryo that's actually going to become the person proper are these three layers in here that you can see. So there's a blue layer, a red layer, and a yellow layer. And those three layers are also depicted down here, albeit they look a little bit different. But this whole layer up here would be the blue layer. This in here would be all kind of the red layer. I don't know why they show sort of a gap in there. But this is basically all red layer in here. And then this layer down here would be that yellow layer. So I mean, it largely matches up. This is a picture from a different book. This is a picture from your actual book. These layers, certainly they have names. So the bottommost layer, the yellow layer, this is the endoderm. And this is going to basically become the inner lining of your digestive tract as well as a couple other visceral organs. So this is like the inside of your gut is the endoderm, which is cool and exciting, but it doesn't do a whole lot for us. The mesoderm, excuse me, is where we find where the tissue or all the cells that will give rise to the tissues that become like your skeleton, your muscles. Most of your other internal tissues, frankly, are going to be coming from mesoderm. But that's still not pertaining to us. That's not where our nervous system comes from either. It's this topmost layer, the ectoderm, where our nervous system is actually derived. So most of the ectoderm, which is what they're kind of showing off to the side, this is kind of clipped, but this would continue off this direction on either side of the slide in blue. Most of that's going to become your integument, so that's going to become your skin. But there are these two little patches. There's this neural plate structure here, and then these two structures that are shown in kind of purple. These are basically going to become neural crest. That's going to collectively give rise to all of our nervous system, both central and peripheral nervous system. The neural plate itself is actually going to give rise to the central nervous system. And then the purple patches will become neural crest, as I'll show you in a sec. And then that gives rise to all of your peripheral nervous system. So here's, this next slide kind of gives you a time lapse of how this process works. And this is happening really early on. This is like week... Oh, I should remember that. I want to say it's before week 5. I mean, so this is really early on. I should have looked it up, but I didn't. I'm sorry. I'm blanking on exact time course, but again, by week 5 is even way too late for all this. So here we are. So at this point, we've got this would be like a top-down view of what was shown on the prior slide. So there's a little bit of like an undulation thing going on in these different layers, but we basically have our 3 layers, our ectoderm, our mesoderm, and our endoderm. And this is shown in cross-section. This is what it would look like if we looked at that embryo from above and we kind of carved away the all the endometrium that's covering over top of it. This is what the embryo would look like. It's just basically this flat layer. It wouldn't have this coloration. This is false color just so we can track the different kinds of tissues. So the green's in the center, the purple's around the periphery, the blue's around here, and all this stuff down here is in layers below what we're seeing from the top. What you can already see starting to happen, though, is there's a little bit of a groove forming here in our neural plate. So the neural plate hasn't really moved much, but as we transition from figure A to figure B, what you're seeing is that there's this groove that's becoming really prominent. It kind of starts near the center and it kind of grows out toward the end, and eventually the entire top of the neural plate becomes this sort of long, fairly deep neural groove. And it basically becomes deeper and deeper and deeper. These walls get higher, the invagination gets deeper until it, instead of looking like this big open space, it would start to look like it was starting to kind of come towards the tops of the walls would kind of grow towards each other. And eventually they don't just grow towards each other, they kind of fuse. And by that point, that's what's happened here. So at this point, the top of the green bit on the left side is fused with the top of the green bit on the right side and formed a little tube instead of a groove. So here we've got, in fact, a neural tube where the two parts of the green neural plate have fused together and created an entire tube. And similarly, up here, this purple structure, this purple set of tissue, has kind of fused together to form kind of a covering over it, but this is going to become a neural crest, and these don't really stay together for very long. Eventually, the two parts of the ectoderm out here that are going to become skin will also fuse with one another. So not here, but by here, we can see the blue is kind of continue to grow over top and fuse together. And now we have, in cross-section, you can see we've got sort of a nice ectoderm across the entire top. That's all going to be skin. We've got this sort of band of neural crest on either side, and we have this nice, well-developed neural tube that runs all the way down the middle. Yeah? No, yeah, so do the cells look like neurons? Not really. At this point, they're not very differentiated. So it would be sort of a collection of just like undifferentiated, they're not mesenchymal cells, but they're like an undifferentiated embryonic cell. But not long. It won't be long. That's a good point. One thing you can see on here is that a lot of these sorts of differentiations. Like the groove kind of starts at the center and then works its way out to the end. And then the neural tube would start at the center of the embryo. This is kind of like halfway down the organism, and then would proceed towards, they're labeling it the rostral end on one side and the caudal end on the other. Caudal refers to tail. You would have heard of like a caudal fin on a fish is its tail fin. So caudal refers to tail. Rostral refers to nose, so it's toward the nose of the organism. You could probably use, I mean, it's kind of analogous in a person. If you've taken anatomy, we could say anything that's like up towards the head, this is going up superiorly or inferiorly, and you could use that terminology. What ends up happening is that the nervous system kind of turns, especially in people. So like we're kind of going up the spinal cord, and then the brain kind of turns off to the side. So rather than deal with the fact that we kind of go up for a while and then we go anteriorly, we just kind of say towards the nose. So everything towards the nose is rostral, and that's usually the sort of the front of the organism, whereas caudal would be down towards the back of the nervous system development. And that's typical terminology that's used here. Here's some pictures, just so you can see this is a real thing. So here is really early on. You've got a developing embryo. This would be the neural plate right there, and it's still pretty much a plate. There's a little bit of a dip, but it's just basically a plate at this point. Here at this point, it's becoming a pretty prominent groove. So that's a nice neural groove. And by this point, you can see we've just started to have the two sides up here. They've started to fuse together up at the top, and now we have a nice, pretty well-developed neural tube. It's probably not done fusing at the top, but it's pretty close. You can't really see the neural crest proper in these pictures, but at least you can make out what's going on with the neural tube pretty well. Again, all of the central nervous system of your body, so the central nervous system, that's your brain, and it's your spinal cord, that's all going to develop from this neural tube structure. Whereas the peripheral nervous system is all going to develop from the neural crest. This slide shows some of the same things. So on the left over here, so again, we've got an undifferentiated neural plate at this point. Here's neural groove. Here's a more prominent neural groove, really prominent neural groove, and here's where finally you're starting to form a neural tube. So we're getting that fusion that's occurring. Another thing that can happen, and it's not comfortable to think about, but this doesn't always, unfortunately, work exactly the way it should. So there are cases where you don't get complete closure of this neural tube. So the neural tube is supposed to go, starts at the center and it's supposed to go all the way rostral and all the way caudal. So here, and this is nice, they're giving me my day prompts here. So we're at 22 days, so we're about 3 weeks in is when this starts. And even by a day later, it's mostly been completed most of the time. And that is the sort of time course that's supposed to happen, but sometimes you don't have complete closure of the neural tube at either the rostral or sometimes at the caudal end. And that can result in substantial birth defects. So if you don't have closure of the neural tube at the caudal end, so down towards the base of the spinal cord, you can run into a condition called spina bifida, and that's what they're trying to illustrate here. This is a picture of actual spina bifida. So this poor baby, so here's what happened. There was not complete closure of the neural tube, and so what you're actually seeing here, this is the dura mater. This is the outside covering of what should be the outside covering of this kid's spinal cord, but it's kind of ballooned outward because there wasn't complete closure of the structure, as should have happened. You can have a similar thing happen. I can't even look at the pictures of it, but you can Google it if you want. But once you're a parent, like, I don't know, I'm sensitive, so I just can't do it. But you can have similar kinds of things happen on the brain end of things. So if there's not complete closure on the rostral end, you can have a condition called anencephaly, where you just don't have full formation and closure of the brain. And usually what happens is it just kind of looks like, part of the kid's brain just didn't, and skull just didn't quite form. I don't usually see the same kind of like ballooning dura mater thing going on. It just kind of looks just not formed. And so that's actually a reasonably accurate depiction of how it might look. Okay, so this gives us, this is like day 22, so we're 3 weeks in. And at this point, we've already created the foundation of the tissues that are going to become all of your central nervous system and also all of your peripheral nervous system, the neural tube and the neural crest. But they're certainly not there yet. We haven't made a brain yet. We just have this tube of tissue, undifferentiated tissue that's running down the sort of the dorsal backside of the organism. So what happens next? So the next steps are differentiation of this neural tube. So here they've kind of done this is like the day 22 picture again, but they've kind of carved away at the top. And what you can see is that you're actually starting to have, even by day 22-ish, you're starting to have a little bit of initial differentiation of the neural tube up in the rostral area, such that there are some little blebs that are sticking off, little vesicles we call them. And these vesicles are actually going to give rise to different brain structures. So they've kind of taken this little thing and they've blown it up here so you can see it better. It's really divided into three major sections right now. There's there's like these little, almost like ear flaps. These are not actually ear flaps, but I don't know how else to describe them. But there's these little flaps or blebs that are kind of sticking out, and then there's sort of a peak to it. This whole area is called the prosencephalon. This is basically going to become all the forebrain. So this is where your cerebrum is. It's where some of your central brain structures like the thalamus are derived from as well. And I've got more on that in future slides, but just to give you a feel for how this is working. This area is referred to as the midbrain, or the mesencephalon. Mesencephalon, so encephalon refers to the head, mes is mid, so this is your midbrain at this point. And the midbrain, we continue to call it the midbrain throughout even into adulthood. But it's not a very large area of the brain, but there are some key important structures that are present in there. All of this most posterior vessel, at least as far as this anterior bit, or rostral bit, this posterior part is called the rhombencephalon, or the hindbrain. And this is where you're going to have your pons, your medulla oblongata, and also the cerebellum is going to be derived from that structure. It doesn't end there, though. So like one thing I think, at least on this part of the picture, they show is that the tube continues on, because that neural tube continues all the way down through here. And the rest of that neural tube is all going to become spinal cord. So your spinal cord basically continues, and it's just a continuous structure that extends from the brain. There's not really a really hard and fast spot where one ends and the other one starts. It's all derived from the same ultimate tissue. And I forgot those words were there on my slide, but there they are. We just said all those things. All right, so let's flash forward a little bit further, because we've only got sort of the very initial inklings of something that resembles something like a brain. So as we continue to see development of these vesicles, we'll see that especially up in the prosencephalon, we see some secondary vesicles developing. So the primary vesicle of this whole blue structure, this was the prosencephalon. That's what's on the prior slide. Secondary vesicles are sort of secondary modifications of this thing. So these two large blebs that are coming off in the most rostral part, those are called the telencephalic vesicles, and that's going to become your telencephalon. Basically, that becomes your cerebrum. So your two cerebral hemispheres are all derived from this area of your forebrain. The central spot that's in the middle in here is your diencephalon. So that's going to become basically your, I skipped one. The diencephalon is going to become your thalamus and your hypothalamus. I think your pineal gland's technically in there too. But these are sort of core central brain areas that are involved in a lot of sensory thought processing and filtering. The hypothalamus, as you may be aware, well, I'm going to get into all this stuff in a second, so I need to stop getting off track. There's also these two little optical vesicles that are shown here, and they're showing very early on, and then they kind of grow out from this spot, and they grow outward and forward. They become an optic stalk and an optic cup, and this basically becomes your retina and your optic nerve. So these will grow forward. So the retina of your eyes is actually brain tissue, and so that's a full process of your brain sticking forward. So I think there's a quote, it's one of the textbooks that I use. As you stare longingly into your partner's eyes, you're actually staring directly into their brain, because the retina, that is actually their brain, or part of their brain tissue that you're seeing there, because it's part of this little optic vesicle that emerged off of this large structure, the prosencephalon. And then, of course, the stalk becomes the optic nerve, which conducts signals from the retina back towards the brain. So what we can see happen, maybe this was the picture from the prior slide, or the state of the prior slide, but the telencephalon just keeps growing. So we have these two sort of smaller telencephalic vesicles, and then they grow back and bigger and bigger and bigger, and they just kind of start covering everything, such that, you know, by the time you get done developing, this is more of like a mouse brain design. But you have these large cerebral hemispheres that are kind of covering over the top of all the, like the diencephalon's kind of buried in there. The mesencephalon you can't even see anymore. This would be the midbrain. It would be kind of like through that telencephalon. Here's the hindbrain back there. At least you can still see it. But the telencephalon just kind of takes over everything. And this is pretty typical for what we see in mammals, and certainly definitely within people. All of the various cerebral structures of the cerebral hemisphere are derived from those telencephalic vesicles. Those include these two bulbs up here. So these are the two olfactory bulbs, which are reasonably well developed in people. These basically sit directly over top of our olfactory mucosa. So like we've got, you know, you've got your nasal passageways. And then you can actually see in the brain, there's this thing called the cribriform plate with a bunch of little holes in it. And there are then axons that travel from our olfactory epithelium, where all of our little nose sensors are, or our nasal sensors, olfactory sensors. And they all course up into the olfactory bulbs, which just like sit right on top of them. And then from there, this is how we sort of get our sense of smell coming in. And that's all emerging from the telencephalon as well. And then we also have, in addition to all the various gray matter, which is, you know, all the, like the synapses and the cell bodies, we have a lot of white matter tracts. So these are collections of axons that develop connections. And so we've got, you know, those are connections within the hemispheres, connections across the hemispheres, and connections where we have axons that travel from the cerebrum down out into the hindbrain and even down to the spinal cord, and vice versa, coming up from spinal cord and up into the telencephalon. Excuse me. So by this point, I've been throwing out lots of structure names, but I think, so it's time to start sort of slicing and dicing, start labeling some things a little better. So here's a slice through the very rostral, most rostral part of the telencephalon. So we've sort of cut here, and then we're just sort of taking that slice and then turning on its side and looking at it from behind, and that's what's depicted here. So this would be the upper surface, dorsal surface. This would be the downward ventral surface over here. So we have these two large telencephalon vesicles. You can see that they are largely hollow, and there are these two large ventricles, we call them, that are contained inside. They're the lateral ventricles. These lateral ventricles feed down into the diencephalon, which is at the center in here. And so there's a third ventricle, we call it. Like the first two ventricles are the two lateral ventricles. We don't call them 1 and 2, though. We just sort of assume that there's two of them. I mean, there are. And then the third one is the one down here. They kind of feed into that one. Here's the diencephalon at the center. In this, over in part C here of the figure, they're showing you some of the major brain structures that are derived from these different structures. So all the telencephalon is basically going to become your cerebral cortex, which is where a lot of the higher order processing of our brain happens. You've also then have the basal telencephalon, which is sort of the deeper layers of the cerebrum. And this is where we see, let's see, yeah, they're not really labeled on this picture, but this is where we see there's a lot of these basal nuclei. So these include the basal ganglia, basal nuclei that are involved in a lot of motor function, the striatum, and some of these other things that we'll talk about later on. In the diencephalon in here, there's these two major structures. There's the thalamus, and we're sort of seeing two sides to it. It's kind of cut in half by the third ventricle. But the thalamus is basically involved in lots of sensory processing. So it's sometimes described as like this major throughput area. In fact, I think this is on the next slide, so I'm going to jump to the next slide. This side of the figure stays intact on the next slide. So this is all the same thing that was on the prior one. So we got our cerebral cortex up here, which is where a higher order brain function is happening. Your thalamus right here. The thalamus is a lot of times, I've said it's involved in sensory processing and filtering. It's a lot of times described as like a gateway to the cerebral cortex. So what will happen is sensory signals will be coming up from your spinal cord, or they can be coming in through any of your other senses even that are coming up through your, from your head. In order for those signals to get to the cerebral cortex, they first are routed to the thalamus, which then receives them in cells there, and does some initial screening, interpretation, filtering, and then will then send those signals on up to the cerebral cortex, where we tend to have more conscious assessment and receipt of those signals. So basically, any kind of sensory information that comes in goes through the thalamus. The hypothalamus is down here. It always seems like kind of a derogatory name, the hypothalamus. It just means it's the thing below the thalamus. But the hypothalamus is critically important. It kind of runs and is like the major decision maker for a lot of the automatic processes in our body. So for example, it controls pretty much all of our autonomic nervous system. And the autonomic nervous system is the part of our nervous system that sends motor output to all of our visceral organs. So this is what's regulating your heart rate and your vasodilation, so your whether or not your blood vessels are dilated. It's controlling function of your digestive tracts. It's doing all these sort of internal functions that we don't have voluntary control over, and it's making most of the higher order decisions, at least, about what to do with those structures. It's also involved in our emotional center, limbic center. It controls a good chunk of our endocrine system also, so our hormonal system is also controlled by the hypothalamus, so it's this incredibly important structure. And it's found also here in the diencephalon. As I mentioned, basal telencephalon, this is where we find the basal nuclei, which are involved in a lot of motor control and motor output, as well as some important structures up in the cerebral cortex. Also, some parts of the limbic system are found in there. This is like our emotional system. Also depicted here is this little band that connects across the two hemispheres. So it's right here. This is the corpus callosum. And the corpus callosum is just basically this huge band of white matter. White matter is basically just a huge mass of axons. So lots and lots of axons. And it's white because these are myelinated axons, and therefore you have rapid signal conduction across them. But the corpus callosum is this large structure where we have axons traveling from the left hemisphere to the right hemisphere and back again. And so that's what we're looking at when you're looking at the corpus callosum, is just all these axons allowing communication between the two hemispheres of the brain. That's forebrain development so far, and major structures of forebrain. We okay? Questions so far? Okay. So if we step back a little bit, going from rostral toward caudal, we get into the next area of brain development. So this is the midbrain. So we're taking a section right here. And so this is showing that section pretty early on in development. It's kind of like that first picture where we have our primary vesicles only, and we're looking at that slice. This is showing it a little bit later, after the telencephalon has had time to grow a little bit more. And there's been further differentiation down here in the midbrain as well, as you can see. Certainly this looks like a ventricle. It's basically a passageway. It's called the cerebral aqueduct. That travels from the third ventricle down to, we'll see, the fourth ventricle in the hindbrain. So it's sort of been walled in and become proportionally smaller over this course. But what you can see is that, at least if we focus on this depiction, we've got a couple different layers, if you will, in the midbrain. We have this upper layer that's called the tectum, and then this lower layer down here that's called the tegmentum. And those are maybe our two major layers. The tectum has two major structures that are important, and one of them is found a little bit more rostral and one's found a little bit more caudal. They are the superior and the inferior colliculus. And when you do the appendix, you'll see better depictions and better pictures of them. Any one slice, you'll tend to only get one because they're found in slightly different spots, moving rostral caudal along the midbrain. They're basically reflex centers, though. So they're in charge of receiving some specific kind of sensory information and then operating reflexes directly associated with that sensory information. So the superior colliculus receives visual information. It gets, there's a good number of axons that are hardwired and go straight from the optic nerves straight over to the superior colliculus. Most of the axons coming from the retina ultimately, through a couple synapses, end up in the back of your cerebral cortex in your occipital lobe, which is sort of like the back part of your brain. But those, there is a sizable proportion, 10% or so, that ends up in the superior colliculus. And what their job is they help to inform all kinds of eye reflexes that we're able to do. So for example, they're helping to coordinate how dilated your pupils are based on the relative amount of ambient light that's present. So as light goes down within an environment, your pupils tend to dilate a little bit, and therefore they can gather more light. They receive more light per unit time, so they become a little bit more sensitive. Conversely, during high intensity light, so if you go out in the daylight and it's really bright, you start squinting, your pupils constrict, and so they're helping to operate that. They can help to operate your eye muscles to allow for tracking motion, for example. So, if I toss an airplane, a paper airplane, across this room, you can kind of follow it. If you hold your head still, you can follow it with your eyes across the room. And that kind of tracking ability is coordinated, at least in part, by the superior colliculus. There's some support of other structures like the cerebellum involved in that too. If there's like flashing light off to one side, you can direct your eyes toward that flashing light. And it's a reflexive action. It's not something where you have to say, oh, I wonder what that, you have to consciously think, I wonder what that flashing light is. Let's turn our eyes in that direction. I mean, you could do that, but you have reflexive attraction towards motion and flashing and other kinds of new events that are happening in your visual field. Again, in part driven by what's going on in the superior colliculus. The inferior colliculus does similar kinds of things, but instead of responding to visual input, it responds to auditory input. So if there's a loud noise to one side of you, you will reflexively turn your head toward that loud noise. And again, some of those types of reflexes are operated with the inferior colliculus, which is found up in this tectum structure, just caudal to the superior colliculus. I mentioned the cerebral aqueduct, so this is part of this network of chambers that's present. That's basically, in adults, that's conducting cerebral spinal fluid around within your body. And then the majority of the mass of the midbrain is composed of a variety of nuclei, so we call them nuclei. I should give you this term. So we've certainly called things nuclei before, like when we were talking about cells, we had a nucleus of a cell. In this case, a nucleus, plural would be nuclei, in brain is basically a collection of gray matter that's not part of the cerebral cortex or part of the cortex of the brain. It's sort of a deeper structure, but it's a little pocket or bead of gray matter that's present within the brain. So it's gray matter, but it's deep. If you had a similar looking thing, but it was in the peripheral nervous system, we call it a ganglion. But in the brain, we call it a nucleus. It's just the terminology we use. So within the tegmentum, there are a number of these kinds of nuclei. The two most important that we'll talk about, one is the substantia nigra. You may have heard of substantia nigra, especially if you've done any investigation into Parkinson's, because these are the cells that ultimately die in Parkinson's disease. So the substantia nigra contains a large number of these dopaminergic neurons. They're neurons that produce dopamine. And that dopamine is sent in axons up into structures within the telencephalon and the basal telencephalon, which are the basal nuclei that help to govern motor control. So, and basically they help to facilitate and potentiate those circuits. So when you lose these cells of the substantia nigra, you lose some of that motor function, and that results in symptoms related to Parkinson's disorder, especially difficulty in initiating actions seems to be directly involved in. The red nucleus is another collection, so we would sort of see, I wish I had I should just get some actual slices through actual brain tissue so you could see these. So the substantia nigra would be sort of a large brownish looking thing, usually in the staining they use. The red nucleus would be kind of a pair of red staining structures. The red nucleus is part of your reticular formation, which actually it starts up here in the midbrain and runs all the way down into the medulla. So it runs all the way down into your hindbrain. But these govern all kinds of things, from sensory filtering to pain route modulation, and the red nucleus is sort of part of those kinds of circuits. Continuing our tour a little bit further. So if we go down into here, we get into the hindbrain. And they show you a couple different slices through that. So here is early on in development, here's later in development. And so they're taking a slice through basically the same area in those two structures. And you can see it does get progressively more differentiated. And then they also, over here, they take a slice at the same stage, but they go more rostrally. And you can see there, again, there's a substantial difference in terms of what you see. Up here towards the rostral end of the hindbrain, you have two major structures that you would slice through if you cut through in this area. One would be the pons. The pons is a major communication area. It's also a major throughput area. One of the big things it does is that it receives sensory information, it receives motor information, and then it relays that up to the cerebellum. And the cerebellum is this larger structure that's sitting up here. This is kind of, still early in development, but the cerebellum becomes the large sort of blob that's sort of at the back end of the hindbrain. Cerebellum's really involved in coordination. So what the cerebellum's job is to do is to receive information from your motor system about what you're trying to do, and information from your sensory system about what's actually happening, and it compares the two, and then issues corrections as needed to make sure what you're trying to do is what is actually happening. That's kind of like the definition of coordination. And it's the cerebellum's job, a big part of the cerebellum's job is to ensure that kind of coordination. The pons is relaying information from the motor system and from the sensory system up into the cerebellum. So that's a big chunk of what it does. And then further back from the pons, which is the more ventral surface, and the cerebellum, which is on top, so if we cut down over here, instead of taking a slice through cerebellum and pons, we'll be cutting through the medulla oblongata at this point. Your book's just calling it medulla, and that's fine. But the medulla is another one of these areas where there's a lot of communication, a lot of signals sent up and down. This is like the base of the hindbrain. So it's getting signals up from the sensory, from the spinal cord. It's sending motor signals down from the higher brain centers down into the spinal cord. And there's also lots of individual little nuclei that control various things. I think I've got some of this information on the next slide too. So let me actually jump to that. So here's sort of summary of these different structures of the hindbrain. So I mentioned the pons. About 90% of all of your descending neurons synapse within the pons. And most of those are going to be then relayed from the cerebral cortex up into the cerebellum. So there's a huge, and others will be relayed and sent down into the spinal cord. But again, there's a large number of these descending synapses, basically going from your motor systems down into the pons, and then you're relaying up into the cerebellum, informing you about what your motor system is trying to do. The cerebellum is then comparing your proprioceptive input, that's sensory input. So this would be like, proprioception is your sense of where your limbs are and what they're doing. So this is kind of like your body position and movement sense. So it's comparing that kind of sensory input to your motor output and then sending corrections as needed to make sure that what you're trying to do is what is actually happening. And then finally, down at the bottom, so here's the medulla at the very bottom. This would be pons, this is cerebellum, this up here would be midbrain in an adult brain. This down here would be your medulla, or your medulla oblongata. In addition to having lots of axons traveling up and down to and from the spinal cord, you have a lot of little brain nuclei that are also present and found within the medulla. So for example, you have a cardiac center. And the cardiac center's job is to directly send signals to the heart to adjust its heart rate. So the hypothalamus we talked about is like the major autonomic control center. It would be making big picture decisions about, okay, given this current state, given how much my body's moving, given my emotional state at this time, this is what my heart rate should be. It then basically tells the medulla that information. The medulla sends the actual signals down to the heart to either increase or decrease your heart rate. You also have vasomotor centers that are present in the medulla that are adjusting how dilated your blood vessels are. You have some digestive centers within the medulla that help to regulate your digestive function. You have all kinds of little reflex centers in the medulla, like you have a coughing center and a vomiting center and a hiccuping center that are found within the medulla as well. Just collectively kind of know there's lots of these vital system nuclei that are operating specific structures within your body. Let's see. I'm kind of out of time, and I want to spend a little more time on these. So we'll have to resume there, but we'll resume there after the exam, obviously. But in the meantime, exam is Monday. If you do have questions as you're studying for the thing, please, drop me a line. Happy to help. I also should be around Monday morning, especially during office hours or 11 to noon. That's a great time to find me. Best of luck, everyone. Have a good weekend.